Morning biologists, in today's lesson we're going to look at humoral immunity. So that is how the body goes and uses cells to bring about long-term immunity and how antibodies and such forth are made. Right, in the following video bits we will go through this in more detail. So in humoral immunity, the first thing that we need to consider is cell-mediated immunity. So cell-mediated immunity is an immune response that does not involve antibodies. Rather, cell-mediated immunity is the activation of phagocytes, antigen-specific cytotoxic T lymphocytes, and the release of various cytokines. So the way that humoral immunity differs from this is that humoral immunity is a type of immunity that will go and produce antibodies. So the antibodies are produced by plasma T cells, which will go and bind to the antigen which is being presented by the pathogen. So the first process that we need to think about in terms of how all of this comes about is antigen presentation in B cells. So we start off with receptor-mediated endocytosis. Is a process of endocytosis which carries the antigen down into the cell. So a specific receptor on the cell surface binds tightly to the extracellular macromolecule, right? So that's the antigen that it recognizes. So the plasma membrane region containing this receptor ligand complex then undergoes endocytosis, forming a transport vesicle containing the receptor antigen complex and excluding all of the other plasma membrane proteins. So a specific receptor on the cell surface binds tightly to the macromolecule. Right? That's what we've just talked about. That's then transported into the cell. Around it, a lysosome will go and fuse with that vesicle. So the lysosome releases digestive enzymes which will go and break the antigens up into smaller fragments. And these antigens are then bound to major histocompatibility complex 2 molecules. So these molecules are then presented to or on the outside membrane of the B cell. Antigen presenting cells specialise in the presentation of antigens to T cells. So if you remember back to when we looked at cell-mediated immunity, we need T cells to become activated in order to go and produce interleukins and cytokines. So the T cell recognises and interacts with the antigen on the major histocompatibility complex, and then that causes the T cell to release interleukins. The interleukins produced by the T helper cell help B cell proliferation. So this is the B cell going and replicating itself. So when we go and we think about this interaction, we start off with the receptors on the surface of the B cells bind to that antigen, or the receptors. So the T cell receptors can only recognize an antigen when it is complex with a major histocompatibility complex. So the T cell receptor binding to the major histocompatibility complex is then involved. Another glycoprotein will then go and bind to it to form a very, very specific bond, and it will only bind to other polypeptide fragments. So the helper T cell then releases interleukins which cause B cells to undergo clonal expansion. So this is where they're going to go and they're going to replicate themselves. So the next bit that we need to think about in terms of it producing antibodies is the process of transcription. So transcription is a process by which the information in a strand of DNA is copied into a new molecule of messenger RNA. The DNA is then safely and stably stored in the genetic material, and it's encased within the nucleus of the cell. So the B cell receptors trigger cell signaling molecules to be released into the cell. Transcription factors are then triggered, which cause the gene that will go and be transcribed into the antibody to go and be transcribed. The DNA in the gene section of the DNA unzips so that both strands separate. One strand is used as a template. Complementary bases then join onto that strand, and this then goes and forms a messenger RNA. So translation is the process that takes the information passed from the DNA in the form of messenger RNA and turns it into a series of amino acids which will finally go and produce the antibodies. So the messenger RNA attaches to the ribosome. The ribosome reads the messenger RNA. The ribosome decodes the messenger RNA in groups of 3, so these are triplicate codons, and complementary bases on transport RNA will go and bind to it. So transport RNA is specific to an amino acid and it can be used more than once. So it's going and is bringing these amino acids to the ribosomes in the correct order. The amino acids are now lined up in the order as given by the instructions in the messenger RNA. Bonds form between the amino acids, forming polypeptide chain, and this polypeptide chain folds up and becomes a specific shape, forming an antibody. So these antibodies then need to be released by the cell, and this is done by the process of exocytosis. So the antibodies are parcelled up in membrane-bound vesicles. These vesicles are transported to the B cell membrane. The vesicle fuses with the membrane and the antibodies are released by exocytosis. So if we were going to go and look at explaining the role of vesicles and the transportation within cells, that is some of the key information that you would need to be thinking about including. You may want to pause the video at this point and just go and produce for yourself a relatively detailed answer to that question. So, humoral immunity is the aspect of immunity that involves the production of antibodies and the production of memory B cells. So, in terms of this process going from start to finish, we start off with macrophages interact with T helper cells which activate them. T helper cells stimulate a specific B cell, so only one will be useful, and that will be the one that makes the antibodies that will bind to that specific antigen. Interleukin is produced by the T helper cell, which stimulates the B cells, causing it to undergo mitosis and clonal expansion. So once the proper B cell is found, like I said, it will go and copy many clones of itself. Each clone B cell then undergoes a maturing process, and the majority of them will become plasma B cells, which will go and produce antibodies. So the plasma cells produce the antibodies. Each antibody is a protein formed in a very, very specific shape and will bind to an antigen. The shape of the binding site is the same as the shape of the receptor site on the cell surface of the B cell. So that's the same as the site that is bound to that antigen. So these antibodies are secreted in large amounts into the bloodstream, where they attach to specific antigens and can either immobilize the pathogen or mark it for destruction. Once the infection has been dealt with, many of these cells die off. Some remain, however, producing more antibodies, but will eventually fade away. Some remain behind and form memory B cells. So these cells form part of the long-term immune system and will be activated the next time that they come into contact with that specific antigen. So hopefully that goes and summarises all of the key things that you need to know about humoral immunity. If you have any questions, then please get in touch.